Hi, hello, and welcome again. Microbehunter here. Welcome to another Saturday live stream. Today, it's again the topic water bears, but specifically, I would like to show you how to construct a so-called a Baerman funnel. I have been somewhat successful with that. I would like to explain to you what that actually is and how you can use it to catch water bears and also other microorganisms. Yeah, at the beginning, again, a little bit of what I call housekeeping. So the sound hopefully does work. Yes, thank you very much. The video also works. I actually just a few minutes before the start of this live stream, I again had some camera issues, but I think I got that sorted out, luckily. Yeah, and basically also this seems to work here. Also the camera here at the top, that's also important because I'm not only going to show you a Baerman funnel, whatever that is, you'll find out, but also I've been experimenting around also with some other things here that I would like to share with you. Okay, yeah, what you've seen here already hidden behind this particle here, that is already a water bear. It's a live view of a water bear, and I'm just gonna show it to you now just in case I'm not able to find one later. Yeah, but there are plenty of other organisms here on the slide as well, and yeah, a lot of nematode worms and so on. Lots of particles here, right? Yeah, occasionally some ciliates. Some single-celled microbes. I don't know where they are right now, but yeah, here, for example, this one here, a pretty big nematode worm. So there are plenty of things that you're able to catch with this Baerman funnel, named according to Gustav Baerman. And here, that's the shell of an amoeba here. That's, of course, a nematode worm. So there are interesting things that you're able to find with that because I did collect a moss sample and I extracted all of these things out of that. So what I would like to do first is, of course, hello all around the world. If you have any questions that are related to this topic, please feel free and post them in the chat. Again, after approximately 1 hour, I'm going to collectively try to answer the questions, at least those that are off topic. Please also include messages or comments or questions to me. Please write at Oliver or at Microbehunter because then they are easier for me to find. Okay, yeah. Very quickly, hello from basically south coast of Ireland. From Vietnam, from Connecticut, the United States, Belgium, from London, Netherlands, northern France, Moscow, England. Okay, yeah, and yeah, I hope that more people, most certainly more people, are going to be joining in. What I will show you now is what I've made here. It's not the most perfect design what I have here, but I just would like to show you what it is. Okay, and this is basically what you have over here. Now, see, that's the problem that I have now, is it's the top view. I'm going to put some of the things here to the side here. I'm going to quickly show you the side view. Okay, so this here is a plastic funnel. Yeah, you can take it out. It looks like this, right? And in this plastic funnel, what I have here is a sieve. So it looks like this here, right? Yeah. So there's it's actually used for. Yeah, I don't know if you want to separate tea leaves or things like that. Yeah, and there was also a handle on here which I cut off, right, so that I can put the sieve direct in here. And in it I put some moss, right, and it filled up everything with water, right. And you can see already here that the particles that also other organisms that basically are in the moss, they are basically go into the water and then they will fall down, yeah, through the funnel. And they basically fall down. The sieve will kind of retain the moss. Yeah, some people also use soil samples. Yeah, and then the particles and the tiny worms and a lot of rotifers and so on, they will fall down here. And on the bottom here, I've got this here is supposed to be a rubber hose. It's not. I simply used a disposable pipette. Yeah, so I used one of these things here. Right, it was very flexible, and I connected it down here with so-called parafilm. I'll show you what that is, and then I used some clamps to hold it together, so to prevent the water from flowing out. And you can already see here, it's a little, yeah, these are where all of the particles are. And already today, a couple of hours ago, I've actually collected already a few samples here, and yeah. I'm going to also collect some more samples. And this is actually quite a nice specimen to put under the microscope because it not only contains a lot of microorganisms, but of course, as I already mentioned, water bears, if you're lucky, lots of nematode worms and other things. So this is actually a very effective method of concentrating living things that can be found in moss. The Baerman funnel was developed in 1917, and so it's over 100 years old already. And Mr. Baerman, he was a parasitologist, so he studied parasites, and he used this method to collect samples from soil and from other decomposing leaves, maybe of solid material in any case. And it's still used today. And as a matter of fact, if you just Google online or if you on YouTube, you can find actually several tutorials on how to set up those. Okay, yeah. So a couple of other things here. Today we're hunting a big game. Yeah, hello from the United States. Okay, and I didn't know parafilm was watertight. Sure it is. So at least I've been using it. I will show you what parafilm is. It's, I think, the most interesting invention ever. Yeah, so this is basically parafilm. I'll just show it to you. This is parafilm, and this parafilm can be used for a variety of different purposes. And what I've done is I can just show it to you because this is really useful, and this is how I actually connected the things together. You have to cut it here so that the brand name is parafilm. Yeah, and when you basically do the following, then you can actually you have to first separate the paper here, and then you can get watertight and also airtight seals, and just show it to you. No, I'm going to show you not. I cut it the wrong way. I'm going to show you something different. Look, because a more useful way. You cut it square. Now you have to be a little bit patient. You have to let me stretch it maybe a little bit here. Yes, maybe that works better if you stretch it. You have to separate it from the paper. And I'm a little bit see, usually it goes very easily, but when you have a demonstration, you always have the so-called demonstration effect. Slowly, and especially when I'm stressed, things don't work quite the way that they should work. Yep, here we go. Okay, here we go. Okay, so this here is the parafilm, and I'm going to show you now something what it's used for as well. Let's make this a little bit bigger, right? So I have my water sample over here, and it's there's a danger of it evaporating. So what you do is you take the parafilm and you can stretch it, and as it stretches it starts to stick. Yeah, and look at this. Yeah, this is gives you now a very good seal, and it's able to close it off. Yeah, so this is basically what I've been using it for as well. So not a sponsored video, by the way, just showing you what I've been using here. And I used this parafilm also over here to connect the pipette here. I cut it off, this one off, and I inserted it, and I basically wrap the parafilm around here to give it an airtight, especially a watertight connection. You can already see that there's still some more of these particles floating down here. So I buy it cheap in bulk on AliExpress. So in any case, this is what I've been improvising a little bit. So this is again not the best solution, but what I would like to do now is I'm going to show you now how you can collect some liquid from in here, and I'm going to do that by just a second. Here I've got a beaker, and now this is a little bit the problem with this setup, that there is not enough space here. So what I have to do is I have to kind of lift this up here. Okay, I'm going to lift it out. I'm going to keep it closed. I need to lift it out. Okay, because there is of course a lot of water in here. And I'm going to remove now the clamps, holding it close, and look what happens now. Okay, this is highly improvised, okay, but you can see all of those particles in here, right? Okay, and that's it. Okay, and this stuff here is hopefully contains now a lot of these interesting, hopefully interesting organisms. So I'm clamping this one off again. This is not the way it's supposed to be, but I'll be honest with you, I had problems finding sufficiently flexible tubing. Yeah, so that's why I kind of improvised it a little bit. Okay, so here we go again. Yeah, so yeah, it's a little bit a mess, but this is actually what we want, right? So there are, of course, lots of also soil particles and things that have come off, and usually water bears and other microorganisms, they like to be there where there are solid material. Especially the water bears like to cling to solid material, right? So then, yeah, here that's basically the one that I collected before, right? Then what you want to do probably is you want to wait until all of these particles kind of collected in the corner over here, then you need a pipette. Okay, so what I've done now is the following: I've got a pipette here, and there is now something that I found out, and it's also something I would like to share with you. And that is that the opening of the tip is a little bit too narrow. So this can be seen as an advantage because then all of those solid particles are not being sucked in. But sometimes water bears and so on, they like to cling to solid objects. So for this reason, I usually cut it off. Okay, so and yeah, then you do the following: you kind of wait until everything settles down. You prepare a microscope slide. Yeah, yeah, yeah. Hello from Belgium. Just yeah, and yeah. Let's and then honestly, things like this are always a little bit a question of patience and a question of luck. Okay, so usually today in the afternoon during the first on the first slide I could not find any water bears, but then on the second try I did again. So usually it was 50% successful. Yeah, one of the water bears was actually also quite big. Yeah, and yeah, we're going to wait until everything settled down, and then I quickly take up some of this material. Then I wait until it again settles down in the pipette a little bit, and then you can, but that's not very much really, so I'm going to try it again. You want to spread it apart a little bit, and then what I'm going to do now is I'm going to, without a cover glass, the reason why I'm not using a cover glass, without a cover glass, I would like to put it now on the microscope slide and on the microscope stage rather, and I'm going to change over right away. So that is the one that from before. The water bear has, I don't know, disappeared now. Here is the nematode worm from before again. Okay, so I'm going to now put on the slide. You just check what's happening right at the top here. You can actually see me inserting the second slide here. Problem is that I spilled some water and now it starts to stick on the microscope stage, so I have to wipe it a bit here. You know what? I'll just remove the other one first. Okay, so at least, even if we're not lucky now, at least we've seen a tardigrade before, and then here we go. Okay, and then we start again with a low magnification here. Okay, and then let's see what we're able to see here, right? So that's now my 4 times. Yeah, and then usually what I do is I scan the whole slide here, and then maybe if lots of rotifers, yeah, and then we're able to have a closer look of what we're able to find here. Yeah? So this is still, I think, a little bit small, but I'm a little bit interested in this guy over here. Do you see this arrow? I don't know this. Let's have a look what this is. Okay. So I'm going to move this one into the center. Right. Let's go up with the magnification. I flip in the condenser. And yep, here it is. Okay, here we already have the next moss piglet, the next water bear, nematode worm over here. Yeah, some single-celled microorganisms over here. Another nematode worm. Look, You see that a moss is a very yeah, active. Yeah. So you see that actually we were quite fast in finding them. Yeah. Here it is again. Okay. I also see a bear skin rug further down. Yeah. So I think you get the idea, right? So this is a little bit the nice thing here because it allows you to really concentrate, yeah, a lot of samples. Yeah. So and that basically, I could now say everything I've shown to you, everything that I wanted to show to you, and now we're finished with the live stream. No, and it's not the case because I actually did prepare something else, a little bit of a proof of concept that I would like to share with you, and it's not working yet perfectly. Okay, so it's I just want to show and share with you something just as a little, how do you say, as a little yeah, proof of concept, and that is something that I've been working on today, and I kind of got it to work 30%. Yeah, I found an interesting sun shape above. I don't. Did you see something here? If you find something that's shaped like a sun, then it's a heliozoan. Yeah. So, but what I would like to do now is I would like to show you now something else that I've made, and now watch this. What I have at the top. Yeah? Watch the stage. Watch the stage. Watch the stage, because I'm going to move something in now. I'm going to move something in now into the stage. Now here it is, here it is, here it is, here it is. What do you see? Does this look a little bit like a highly improvised micro manipulator? We've been talking about this already. So what is, I need to lower it down now. Okay, I need to lower it a little bit. Yeah, it's a little bit too high up yet still. Okay, here we go. Yeah, so, and this is a little pipette, and I will show you how it works. It's actually surprisingly easy, and it does work a little bit, okay? So I'm gonna show you all of the advantages and disadvantages. I'm now going to move it into the water droplet, and you might also see how it approaches, goes in here, okay? Let's see, where is it, 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 up? Is it, here it is, okay? Yeah, you see actually now the tip of the pipette, and look, I can now move the stage, right? And I'm able to do now something crazy, and I wonder if it works. I can move right next. I move right next to the water bear, which is hiding somewhere in here, I guess. Here it is, and I'm trying. Okay, look what I have here. It's an air tube from an aquarium, and now I'm going to try to suck it in. Okay? Up. Okay, I sucked it in now. No. Look at this down here. Do you know what this is down here? This looks like the skin of a water bear which molted. Okay? So this is basically the thing. Then I try to move over to a place where there is less water, and then I try to release it. Okay, is this the water bear? I don't know. No. Yeah, so that's kind of the idea. It's go down to a lower magnification, and I don't know if I actually caught it or if I was able to transfer it or not. Yeah. But then I'll show you how I've made this thing. This is a little bit of a proof of concept, okay? I don't know. Maybe I didn't suck it in. I don't know. Yeah, I'd have to try again. Yeah. So I think I'm just gonna move over. But you get the idea, right? You can basically position this little tip wherever you want to have it. I don't know, whatever I want to suck in here. I don't know. I mean, you get the idea. Whatever it is that you, maybe a little worm here. Okay, so, and suck it in. The worm not. Okay, I hold it yeah, tight, and I just go somewhere else, and then. Yeah, and then you're able to transfer whatever you've got. Okay, yeah, this is kind of the idea that I have. Okay, but yeah, I would like to quickly show you in any case how you can can do that, and how I've set the whole thing up. And this was a little bit something that I was kind of experimenting around a little bit over the last couple of hours. And yeah, one possibility could of course be that you don't use the setup that I have, but that you try to pick it up with a pipette. It's also kind of works, but you need a very steady hand in order to do that. But the real big question is how can you actually make the tip much thinner than it actually is? And that's also something that I would like to show you, okay? And then whether or not you want to play around with a tube, an air tube or not, or whether you want to use a pipette, or whether you want to use something, maybe a disposable one, that's in a different question, okay? So, you could also use eyelashes, right? I heard that the way of positioning diatoms, great pieces of art. Are there any more ways or applications? Yes, in the past, people have used their eyelashes to pick up individual diatom shells and position them. However, this would not work here, because here it's surrounded by water. So if I were to insert an eyelash into the water, the things would not stick to the eyelash. But the diatom shells were moved around with eyelashes indeed, because the eyelashes were a little bit oily and greasy, and this was allowed them to pick up the specimens. Yeah, yeah. So this is So you see that when I move it around, the pipette is a little bit flexible as well, right? So this is a little bit the thing that I've been playing around with, and I just wanted to share it with you. But I'm going to show you now how I've actually. Well, I was able to pick up the nematode here, after all. Yeah, I'm going to now show you how you can actually make the tip very thin, okay? So, and in order to do that, I would like to simply show you a comparison now. So what you see over here is now the thin tip, okay? But I've got here now the original one, and I'm going to move it also into here so that you simply see the difference in diameter. I have to just see, you see? Yeah, that is the tip of the original one, and you see that it's much thicker, right? And I will tell you what the problem is if the diameter is too thick. It's not only that you're sucking in too much, that's not so much the issue. Most likely you're not able to suck in anything at all, and the reason is that if half of the opening of the tip is not covered with water in air, then you're not able to suck in any water, right? So that's a little bit the problem. If your tip is this huge, then everything has to be covered in water so that you're actually able to suck in the liquid, and therefore the droplet of water has to be very large. So you have to make it smaller, and then also the droplet can be much smaller. Okay, so what is holding the pipette in the air exactly? So that's the next question, okay? I would like to show you over here a big view, and I'm going to move over the camera now to show you how I actually connected the pipette. So what I have over here, all the way down here, I'm going to show you how to make it very thin, right? Then I have a part of a more normal pipette, and I'm going to move over the camera. Okay, what do you see over here? Guess what this is? It's parafilm. Right? That's why I showed it to you. I stabilized the whole thing here with some wooden sticks. Okay, here's some wooden sticks. This is the tube, right, that I connected with parafilm to the pipette tip, right? Some wooden stick for stability, otherwise it's too flexible. And now what's kind of holding it up in the air? Yeah, basically, look what you see over here. You see my computer screen here. This one over here, right? That is I'm going to move it away. That here, yeah, that is a microphone stand or a camera stand. Yeah, so that's basically adjust any kind of stand that you have, and I just used my camera stand and I connected this here, and to that, I connected then here at the top, this contraption that I made, and over here there is this air tube, right, and over here, you can just basically swing it over like this, right, and then you're able to position it directly. Yeah, you're able to position it directly over here, directly. on the stage. Yeah, the camera is switched now back to black and white because this camera will adjust itself automatically. Yeah, so this is a little bit the system that I used. Over here on the desk view, yeah, look, I also used, I only had small pieces of this air tubing, and I also connected it together using parafilm and also some pipette tips that I melted together. Okay, so it says it looks like a chopstick. It's not a chopstick. So what I used is I used these sticks here that are used for if you, for example, want to put meat on here for yeah, I don't know, some chicken or some meat for cooking. So that's actually made of bamboo, and they're quite solid. Yeah. So let me quickly read some of the questions here. Maybe seal off the sticky end of the tube and fold it up to push the air out. Then release the fold to give the amount of suckage defined by the amount of folding. I understand exactly what you mean, and I tried that already, and it didn't work. At least it didn't work all the time. Just want to show it to you. Okay. So what we have over here is yeah, kind of let's move it in again a little bit more. Okay. Every time when you see something like this, then this basically means then it's not completely in water, so you have to add a little bit more water. I will do that, and I'm going to show you a little bit also something that's better. And the idea was the following. The idea was to kind of seal off this end of the tube. Yeah, and very tightly, and then kind of by trying to bend it, yeah, that you're able to somehow change the volume, yeah. You have to press it, yeah, because, and then this way that you're able to suck it in somehow. Actually, now it does work. Before it didn't work. Maybe it was not quite airtight. Okay, so that's actually also a possibility. Yeah, you move it over, and then that's over here. I try to release it again. I push it again. Here we go, and then you have to release it. Yeah. So that is the thing here. Yeah, so I was able to release it again. Yeah, you see over here there are not many. There's this worm here that I got. Now, why would you want to do that? Actually, why would you want to transfer it? And I usually transfer organisms this way because I want to remove all of the remaining particles that you see, just to get a nicer video. Yeah, so, for example, when I made videos of tardigrades and so on, I tried to pick it out individually to get all of the particles removed, so that you only have the organism that I want to show. Okay. Yeah, a skewer. Yes, that's how it's called. These sticks are called skewers. Okay. Yeah, for putting food, I don't know, vegetables and meat, and then you can put it on. And you can put it on a barbecue. Yeah. So, do you have any bamboo slides? Nope, I do not. Okay. Maybe you can use an injection needle as a tip. Injection needle. I never tried it. Okay. I don't know. Yeah. So what I would like to do now is I would like to show you now how I've and this was also not easy, so I also had to experiment a little bit. I want to show you now how I made those thin tips. Okay. Yeah. So for those of you who just joined, very briefly, just a little summary of what I've been talking about. This is a so-called a Baerman funnel, not bear like the water bear, but named according to Mr. Baerman. And there is a sieve in here. I put some moss in here, and all of those particles that are falling down here, yeah, basically you can put under the microscope, and a lot of nematode worms, rotifers, water bears, whatever, will collect down here. And this system down here definitely needs improvement. Yeah. So maybe not make it, maybe a shorter funnel, and but this one is not ideal. Obviously, yeah. So I'm going to show you now how I've been experimenting around. Let me put this away again, the tubing. How to make thin, how do you call this, pipette tips, okay? And this takes patience, and it doesn't always work on the first try. So these are disposable pipette tips, and this is a so-called a microliter pipette. You can adjust the volume over here, right? So it goes from 5 to 50 microliters. Yeah. So this is the most common one that I use. Yeah. I also have a larger one, but I'm going to use the smaller one. This one goes up to a milliliter. yeah, which is 1,000 microliters. And the question is now is how can you make this the tip here in front? How can you make it smaller? Okay, and I'm going to just show you a possible method which requires a lot of a pretty steady hand, and maybe it's not going to work the first time. Yeah. So and what I do now is I need again. Look what I've got here. You already know that. This is my my alcohol burner, and I just realized that here's the lighter. Again, I have to make sure that this is not, it should be sufficiently far away, that this does not hurt my camera. So let's move it further. Okay. So it's not directly beneath the camera now. Okay, so that's an alcohol burner. And then what you do is you try to melt the plastic over the flame, and then immediately you turn it vertically. Otherwise, that's important. Otherwise, I've found difficulties. But this might not work the first time. Okay, so I'm going to try this a few times maybe. You rotate, 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 and vertically, you hold it vertically, and then you carefully pull it. Actually, it worked. First trial. And then you hold it like this. And finished. Okay. Let's move it a little bit away here. I'm going to put it up here. See? And we're actually already finished with that. Okay. Yeah. Here, this is how it looks like. Yeah, so this is actually much thinner, and then I cut it. Okay, something like, yeah, usually maybe diagonally, right? And this is significantly thinner, of course. The question is now, how can I put it on another one? Well, of course, I could directly put it on here. That's of course the easiest way. Yeah, so that's possible. Yeah. And you could actually directly pick up objects like this, yeah. So there are now a couple of comments here. What about connecting a rocket air blower used to clean dust from photography equipment? I guess you can control the suction better with it. And maybe I never worked, don't know much about that. Yeah. In any case, the volume or the suction is quite small. Yeah? I tried to use this pipette to actually suck out the how do you say, some of the liquid, and sometimes it worked, sometimes it didn't, because the amount is not very much. So yeah, I was not able to get a lot of suction together with that. Yeah. I used the funnel for filtering seawater and sea sand for detection of diatoms, and it works well. Okay, interesting. Baerman funnel for filtering seawater. Huh? Can you tell us what sieve size you're using? Okay, the sieve size, I don't know. This is just a regular kitchen sieve that I'm using here. And I don't know the diameter of this. This is just a, this is a very coarse one, right? So this is not a fine meshed one, right? This is actually pretty large, yeah? As you can see here. This is actually the regular one that I bought in a supermarket for I don't know, if you want to filter tea or if you use some juices or whatever, right? So it's really nothing unusual. I used the plastic one, or maybe it's nylon. I cut off the handles here, and that's it. Yeah. So definitely not suitable for filtering water, because all of the microorganisms and tardigrades and so on are way too small. Yeah. To be held back. So I'm using the sieve here actually only to hold back the larger particles that might be, and sand and small stones and whatever that might be in here, right? So I'm just reading some of the comments again. Yeah. So yeah, I just want to show you now also I put this back in. You see that actually the moss looks kind of green now. I'm just going to show you. I collected it just today in the morning. Right now where I am, it's evening, but I just want to show you now the original, how it looks like originally. This is the original one. Yeah, which I collected. So it's quite dry. and you have to give it a couple of hours maybe for it to soak up sufficient liquid, so that you're able to, so that all of the microorganisms and microanimals and so on, that they kind of come back to life. Yeah? And yeah, so that's basically how it looks. And you know what we're going to do is I'm just going to try to find a few more, and then maybe we're able to find some more things in here. Yeah, I have to say that of course it's possible to also directly rinse out the moss on a petri dish like I've done before many times. But actually, I do get probably better results with that. And one of the reasons is that if you rinse out the moss in a petri dish, you have all of those sand particles as well and all these things, and they're all being held back now by the mesh. Yeah. So I'm just reading again here. Could you use kitchen or filter paper? Not filter paper, because I think filter paper, like for example from a coffee filter, which I've also got there, is definitely too fine meshed, and it's probably going to hold back too many things, right? So that's actually one of the ways why I've been experimenting with this suction device here with a pipette, in order to separate the organisms from the whatever particles might be in there, right? Yeah, because I mean, it's already an improvement what I have here, but if you are then using filter paper, then everything's going to be held back, and you don't see, probably don't see anything. Yeah, yeah. I'm using a 250 micrometer mesh for mine. I also have some 500 micrometer and 850 micrometers. 850 micrometers is almost a millimeter, right? So that's already pretty coarse, and I guess it's fine, because the point is it's supposed to only hold back the, how do you call this, the the sand and the larger particles. Yeah. So let's have a look. I'm going to change this tip here again, and I'm going to now probably remove this here again. Just a second here on the top, and let's give it another try. Okay. So I'm going to cut off this tip again here, so that it's easier for me to take up some of the material here. Okay. It's interesting, even though, okay, now you see it, it's actually quite interesting, even though there's so many solid particles in here, it still won't take it up quite as easily. Okay, now I got more. You want to probably spread it apart. Okay, and then I simply quickly check it, and now I'm going to simply move this thing away here, because I just want to observe if we're able to see anything of interest here. So again, the scope view, the arrow is gone. Yeah. And usually if you do some kind of a time-lapse video, then you're able, as I understand, a Baerman funnel is used for soil samples, where the sample is slightly heated from above to drive organisms down into the soil and into the water. That is one possibility. I've also talked to someone already before, and they did not heat it, but actually they used light. So they had a bright light on top of the funnel, and then some of the organisms actually moved away deeper into the soil or away from the moss or whatever you've got, and then they moved downwards, they reached the water, and then they fell down, right? So that's kind of, they got trapped this way. So you can see that the whole range of different variations here. So it does not have to be heat alone, it can be also different things here. So I need to so this is again, and then you see that's one of the things is how can you actually separate the organisms from all of the other particles. That would be actually the next interesting thing to find out. As you see, there's lots of movement going on here. Yeah, I've also found a couple of mites in my other sample. That's okay, here let's go 20 times, And here is another water bear. Okay, moving along. So generally what I found out, and that's also another recommendation is I know that people like to it's almost like a rite of passage, as somebody once said, is to find the first water bear. If they're too difficult to find, change the moss sample. I collected this moss sample inside a forest. There is also quite a bit of moss growing on the wall and so on. But outside my apartment. But I've actually found that they often do not contain as many water bears as those that you find directly in growing on wood inside the forest. And I think this has to do also a little bit with maybe the temperature differences that there are, because if the moss is very exposed and if there are high temperature fluctuations, I can imagine that maybe there are fewer of them around. Yeah, but usually in the moss in the forest, or honestly, pretty much every time I found a few of them. Right. The light method might be good for secondary separation after the Baerman Funnel. Yeah. So basically, once you've collected it like this, maybe there is a possibility to kind of make them move into a certain direction. Yeah. So this is kind of, So I'm not using a cover glass right now. So that's always also, that's why I cannot go up higher with the magnification. Otherwise, I'm going to touch the water droplet, which I don't want to do. Yeah. Oh, thank you very much for the donation. Okay. Yeah. So thank you very much for the donation. I wonder why some organisms in the soil are light phobic. Well, this is actually nothing unusual. Even fly larvae, maggots, they move away from light, and this I think might have to do with the fact that if they move away from the light, then they move into the soil, right? So this is allows them a little bit to move away from danger, because there where there is light, there is of course also the sun, and where there is the sun, there is of course also exposure to dryness. So by moving away from the light into darkness, you're moving into an area that is most likely, also less exposed to drying up. Yeah, my hypothesis. Okay, my hypothesis. Thank you again very much to Mike. Yeah, I've also mentioned before, is what I do is your donations that I collect, and then I buy microscope slides, which I will show you. I have bought a set of microscope slides already, and I buy other equipment that, like for example, the stand for the Baerman Funnel and so on, that I like to share with you. Okay, so I try to kind of experiment around with things, and then I would like to share my experiences with you. And this basically also means that some of the things that I share with you are not 100% perfect. Like for example, this improvised micro manipulator type thing, right? But this hopefully gives other people a little bit an incentive to also start experimenting. Okay. Literature regarding water bears, die Bärentierchen von Hartmut Greven. It's in German though. Yes. Okay. Thank you very much. Yeah. So there are, quite, especially concerning water bears, they seem to be, quite well liked. Maybe because they kind of look cute. I don't know. But there is quite a bit of literature around them. Yeah. About them. Yeah. Actually, heat might be better for secondary separation. Maybe warm up one side of the slide. Yeah, there are indeed microscope stages out there which can actually be heated. There are heatable stages, I guess, especially if you experiment with crystallization or so, or if you want, then you might need a warm stage. And there are some specialized microscope stages out there as well. Yeah. So this is basically the thing here. Let me quickly check, quickly have a look. Yeah, every time when you want to get a better overview, you might want to go down with the magnification. So I'm going to remove this here again. Yeah, and Another thing that I sometimes recommend is, even if there does not seem to be much going on, make a time-lapse video, and then you're actually going to see how much movement there is. Yeah. What do water bears eat? And basically water bears eat bacteria, small cells, small cell fragments, and so on. Yeah? And basically, I think they're not very selective, as long as it's able to fit into their, if they're able to eat them, the things, then I think they're just going to eat it. Yeah. Yeah. So, how large are water bears in microns? Ooh, there are different sizes. Let me do the following. I have a suggestion. Where's the water bear again? Where is the water bear again? Let me try to find it. Here it is. Okay. We're gonna do the following. Okay, so here is the water bear. And just a second, I'll disappear for 1 second and then I will reappear. So I shall reappear again. And the question is how big are water bears? Okay. And I have over here, what do I have here? This is a micrometer slide, calibration slide. I have to get the direction correct. Now the next thing is I can do the following. I'm going to now turn on the arrow again. Here's an arrow, right? And I'm going to turn on another arrow. Here is the other arrow. Okay, so I've got 2 arrows. So one is one end of the water bear is here, and the other end of the water bear is about here. It's just approximate. Maybe the 2 arrows kind of a little bit. Well, it's maybe a little too far apart. It doesn't matter, right? It's, let's say, from arrow tip to arrow tip, that's the size of a water bear approximately. Okay, where is it? No, that's not it. No, I bumped the stage. Here it is again. Okay, and now let's quickly move over, and I have to find it. Where is it? There is a line. And now I have to find it where it is. So this is, I'm using my 20 times objective. Let's go down. And this is my 20 times objective. And just a second, I need to find something. Okay. Yeah, that's the wrong one here. 0.15 millimeters. One division is 10 micrometers. Okay, so that's so one division is 0.01 millimeters, which is 10 micrometers. So I have to find where are the divisions over here. Here they are. Okay, so let's move in here. 1, 10, 20. So let's get this whole thing into focus. So one division is 10 micrometers. So how many divisions do I have? Okay, let's do the following. So I have got a third arrow here. So that is 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12. So 12 times 10 micro. Well, that's actually just a second. Can this be? One division here, it says, is 10 micrometers, 20 micrometers, 30, 40, 50, 60, 70, 80, 90, 100, 110, maybe 120 micrometers, which is approximately a little bit more than 1/10 of a millimeter, if I did my math correctly. Yeah. So basically, if you are to see it on separately, then it's like a tiny dot. Yeah. 1/10 of a of a millimeter. So that's basically approximately the size. Yeah? So these micrometers. I think that here we are again. So I have to refocus. So lots of things. I personally have to tell you, I'm a little bit surprised always that these critters are able to survive the dry moss. And another thing that I would like to show you, these things here are the so-called the tests, they're called, of amoeba. A test is a shell, like a structure, and this one seems to be an empty test. The test basically from testate amoebae. These are amoebae, that basically have this shell around it. You changed your magnification before counting the length of the water bear. Surely it has to be the same. Yes, I changed it and I changed it back. What I've done is correct. I did change it first to the lowest magnification so that I'm able to center the slide, and then I switched up again. Yeah. So this is the thing that I've done. So when you, for example, I don't even know where it is. Yeah, here, for example, I've this one is now first was at the lowest magnification, and then later on I switched up again. Okay. Yeah. So it has to be obviously the same magnification, otherwise it's not going to work. Yeah, 120 micrometers to 660 micrometers. Yeah, so essentially I had 120 micrometers. Thank you for the research. So you see that with this one, I was in the lower, at the lower end. Yeah. So that's basically the thing here. Always, so in case you're kind of wondering what I'm doing, I like to simply do a little bit of exploring this way. Let me quickly switch off the arrows again, because they're kind of disturbing. So that's usually what I do. I collect some samples and then I simply have a look at them, hoping to find something more or less interesting. Yeah? Again, it would be the next interesting thing would be how are you able to separate all of the moss particles, all of these things that you see here from the rest. Yeah? And I've also seen some diatoms even in the sample. So there's quite a few things here. So what time is it? Okay, 1:52. So basically, what I would suggest here, let's go back a little bit to the Baerman funnel. I'm struggling with the air tube here. So let's put this away. Okay, let's move it out of the way. Well, I would actually propose a couple of changes to the system that I have here. Okay. Basically what I have is the following. I have down here as I have to change to the desk view. Okay, yeah. So I would, if you want to do something like this, I do have a couple of. I propose a couple of changes here. First of all, I think the funnel is too long. Okay, this has to be shorter, and it would be nice to somehow have a system here where I could actually turn it so that I'm able to let down some of the liquid. So this is a little bit the next thing that I'm kind of trying to experiment around with, so that I'm able to put a beaker like this here, okay, beneath the funnel, and then somehow either by pushing something or by, I don't know, releasing a clamp, then I'm able to directly collect the sample in a beaker. This system right now is just not satisfactory, obviously. So that is something that I'm kind of trying to work on as a next option, because when the whole thing is optimized, then of course you can use this to collect microorganisms also from soil, from other matter as well. Yeah? Now, if you just as a short, how do you say, as a short suggestion, if you have don't want to go all the way with the funnel, I quickly show you an alternative method. What you need is some kind of a Petri dish, and then what you do is the following: you add a little bit of water to the Petri dish. Yeah. You take some of the moss, and you make sure, of course, that the moss has to be moist. For a couple of hours, you have to put the moss into water for a few hours, so that the water bears or microorganisms, rotifers, whatever, basically come back to life, and then that's basically what you do. You can already see that there are also a lot of particles here. And then you just try to hold it like this. You wait until whatever is in here collects here at the on the bottom, and then you take that. That is kind of the easy or the simple way. also works, but indeed, I think with the Baerman funnel, I'm seeing more a higher density of organisms. Yeah, yeah. Maybe a separatory funnel the chemists use. They're of glass. Yeah, that would be a possible. I see what you mean. Okay, yeah, that's the one that from a separation. Yeah, when you're that might be a possibility. Maybe a small one. Yeah, a small use the small finger of a rubber glove. Oh, that's also an interesting thing. The small finger of a okay, maybe you put some kind of a yeah, yeah, the finger of a rubber glove, and then this is flexible enough that you can actually press it and close it off. Huh? Why can't we just use a funnel with a filter? Well, if you use a filter, just going to show you the following. Look, where is it? I have it somewhere again. Where is my filter? Just a second. Yeah, here. Look, if you want to use filters, like coffee filters here, then I think that they are too dense. Okay, so I don't know if the microorganisms or the water bears are actually able to go through the filter. So that would be a little bit my concern here, right? That you're essentially filtering away too much. Yeah. So I mean, what we can try is, yeah, we can also try to put something from here under the microscope. Let me quickly put the micrometer slide away, and I have to put it into the box. Okay, and yeah, I'll just take. Yeah, but you see there is not a lot here, so you know what? I do the following. I take something here. Okay. Yeah, again, I do not have high expectations now, okay? This is again just a little bit of a proof of concept, yeah? And then I go here, look. And I put it directly on the slide, okay? And that's basically what I've washed out, yeah? Just now. Yeah, you get the idea. Yeah, next possibility would be an enrichment culture. Basically, add a little bit of food, and then you kind of hope that these, those microorganisms are going to multiply. Yeah. I mean, using a coarse filter like the one you have. You mean why can't we use a funnel with a filter instead of the entire Baerman funnel? Just use a funnel with a filter, coarse filter. Don't entirely know what you mean. Are you talking about the sieve that I have over here? Yeah, the sieve itself. Yeah. Okay. Maybe this is what you mean. Look, I just show you. Maybe you mean something like this. That should maybe also work. Look, why not try it out? Why use a whole funnel if, look, this is going to be messy. Why not use a full, if you can also try something like this? Okay. Yeah. I think one of the reasons why also in the sciences and research and so on, why they use Baerman funnels is because it is simply a standardized procedure. Huh? But I get it, of course, that this way you're also able to get out particles here. Yeah. So that's just saying. There were a couple of questions. One of the questions that I think I just read is the following: is where do I get the arrows from? Who asked that? Yeah, Mike was asking this. Where do you get the arrows? So just show you. I have to go to the scope view again here. Yeah, I've got some arrows over here. Yeah, that I can move around. And the question is where I made them. So basically, I'm using OBS Studio, and I can superimpose images, right? So these are not the standard Windows arrows, but I kind of designed it, and I can do all sorts of crazy things with them. Look at this. I can change the size and whatever, right? So this is basically nothing more than some superimposed graphic. Yeah, and I can copy paste them, and I'm able to therefore superimpose whatever I want to do. Yeah? So just reading again. Okay, but there seem to be no more questions. In an aquarium shops, you might find valves. Yes, as a matter of fact, look what I've got here. And I just also show this to you. And where is the desk view? Now, these actually are some of those things here that you can connect. You can connect those tubes. So I'm experimenting, right? And there are probably a variety of different ways of how you can solve that problem. Yeah, it's a more how do you say a question of trial and error. Yeah. So separating funnel that the chemists use. Okay. What is the product name of those clamps you use? They're very handy. Ah, like the clamps? Yes, as a matter. You mean these clamps here, right? Yeah. I don't know. I don't know what they're called, honestly. Yeah, I found them in a paper shop, and they're actually used to clamp paper together and things like this, right? And actually, I will tell you actually why I bought those. Okay, I will show you why. I'm going to quickly do the following. This is a little bit off topic of the Baerman funnel, but it is microscopy related nevertheless. The reason why I wanted to use those is the following, because I wanted to clamp two slides together like this. Okay, you clamp them together like this. You put a string on here and you hang it into a pond for a couple of weeks. Okay. Why would you do that? Because then you will have a biofilm forming on this glass slide. Okay, so algae growing, microorganisms, stentors, maybe you watched my previous video, Vorticella, I don't know. Yeah, and then because it's clamped together, the biofilm is only going to grow on one side, and then you can take it off and you can put it under the microscope. Right, and the bottom side will be hopefully free and will not have a biofilm because the other slide was there, right? So this is basically the reason why I bought those. Okay, those clamps here. So a reason number two where they are used is the following: when you are actually preparing insects, if you want to make then you do the following. I'll also show you how this works. What you do is you take two cover glasses, one on the left here, one on the right here. I don't have an insect right now that I can show you. You put the insect, in here, after it's been treated, and then you put the second slide on top. Thank you very much. The questions arrived. Here are your questions again. I'll put them over here. You clamp it together like this. Okay, you clamp it together and then you submerge the whole thing in alcohol to remove the water. Yeah, right. And then the insect is actually really squeezed flat, but you put it into alcohol for several hours, and then basically you can make a permanent slide. So that's actually, these are some ways of how this can be used. Yeah. So this is so I consider them to be quite useful. I don't know if they're to what extent they are how do you say, stainless or not, or whether they're going to start rusting. I don't know about that. Yeah, but that is actually the reason why I bought them. And so you see that these are some of the other projects that I'm kind of planning out or experimenting out with. But the first thing I'd like to do is I would like to just clamp them together and attach a string here, and then hold it in some water for a couple of weeks maybe. Yeah. So that's the reason why biofilms, because of course you can try to scratch material off a surface, but then you're destroying the biofilm. Okay. So what I'm going to do now quickly is the following. I'll be honest with you, I'm going to quickly go through a couple of questions, and then at the end of this video, I do have an interesting water sample standing next to me, which I did not look at yet. And I have no idea what to expect, and I would like to do that with you later as well, okay? So what I'm going to do now is How much percent alcohol? 96%. That's the cheap one. Just rubbing alcohol for dehydration. Okay, so you just use concentrated alcohol. Usually it's 96%. Everything higher is way more expensive and not necessary. Yeah, so I'm quickly going to go through some of your questions here. Maybe I already answered some of them. Okay. I see these are not only questions, but also other comments here. So I quickly scan through them. That's the pear film. AliExpress is always a good source. Okay. I've also bought already several times before from AliExpress. This is in China. And yeah, essentially they send it over and sometimes quite cheaply. Okay. Not relevant so much. Yeah, pear film allows oxygen and carbon dioxide to exchange, but larger things like water cannot get through. So the sample can breathe even without evaporating and without getting contaminated. Oh, thank you for this information. Yeah, so pear film, yeah, is therefore quite, yeah, but you have to be careful because I think it does not withstand some of the solvents. Yeah? So is there only one filtering stage, not two? Well, actually, for the Baerman funnel, it's actually not even filtering really. It just basically holds back the larger particles. Otherwise, you've got some leaves of the moss and so on falling down and so on, and that would be a little bit too disturbing. Okay. Filtering sea sand and seawater. I noticed that when you handle moss and other things with your bare hands, just an interesting notice. Yes, I have no problems with that. Yeah, I have absolutely no problems handling moss with my bare hands. Why should it be a problem? If I'm barefoot, I also walk over grass and over moss. That is not my concern. Things only start to become problematic when things start to decompose and start to turn bad, especially if, let's say, dead animals, meat, things like this. You don't want to touch that because bacteria and fungi that are able to grow on dead animals, they can potentially also grow on you, and that's something you don't want, right? But first of all, moss itself is not something that I'm really concerned about, right? It's yeah, it's not dirty as such, right? So only when you actually growing bacteria, yeah, at a larger scale, then that's an issue. Yeah. Yeah, just up on the left. Okay, there's an egg there. Okay, I kind of overlooked that. Okay, what filter size are you using? I don't know the filter size. It's just a fairly coarse filter. It's not a filter even. It's just a sieve, really, to hold back the larger particles. Yeah. I got the same rubber pipette tips for the same reason. Okay. Yeah. Could you also use eyelashes? I talked about that. Probably not suitable for observing. Not suitable for observing. Not observing, but for picking up things in water. Okay. If the pipette is close to the object glass, only water will be sucked in. Okay, that's correct. It's got to be, the pipette has to be completely submerged here. Okay. Connect the pipette to the micromanipulator hose. Yes, this is what I have done. Okay, you know what I'm going to do? Why not? Because I've already demonstrated it to you. I'm just going to take the, how do you say? I'm just going to show you how I made that. Okay, so this is basically how it looks like. This is how I clamped it, yeah, to the stand. Over here, this is pear film. This is a pipette tip that I cut off, and this is the small pipette that I made using the fire. And here I simply connected the tube with a pipette. I'm just going to show this to you on the other end here, right? So over here, this is the other end, yeah, and what I've done is I've done the following, kind of try to squeeze it in here somehow, right? I try to squeeze it in here somehow, and then I have wrapped some pear film around it, and I cannot do that now, but you get the idea, right? Yeah, so this is basically how I've done this. I pushed it in here, or you can warm it up also over the flame and make it a little bit softer, then it goes in better, and then pear film to make it airtight. And this is what I have done over here. You might wonder why these strange things here. Well, because yeah, I only had short tubes, and I kind of connected together. So what I've done here is the following. I can actually just for the fun of it, because I just want to show this to you. It only takes a minute or so. Always be careful with fire. Okay, look, I melted it and I glued it together like this. Okay, yeah. Then parafilm around it to really make sure it's airtight. And I basically cut this off, left and right, left, right, and I basically inserted this into the tube here, left and right, and then a parafilm around it, and that's how I made those extensions. Yeah, so just a little tip here. It is, as I said, it's highly improvised. Yeah, but I think this here is how to make the tip thinner. I think that's the how do you say the more important part here. Okay, so I'll quickly move on here. So what else do I have? Yeah, that's a stretched tip by heating. I thought it was the rubber micro tips I bought. No, it's actually just the disposable plastic tips that I have. Yeah. Okay, what's the holding? That was my microphone stand. Maybe seal off the sucky end of the tube and fold it up to push air out. Yes, I tried that. Yeah. What about connecting a rocket air blower? Never tried that. Yeah, talked about this as well. I believe you can use a capillary tube that glass and then can be heated and to pull apart to make a small tip or manipulator. Yes, and I have actually done this as well. You can actually use a glass pipette and you can pull it apart as well. I think it's even easier with glass than with a plastic, but I didn't have one right now, and I simply wanted to use the plastic ones because they were easy to connect also to the pipette. Yeah. I used a funnel for filtering sieve. Okay, we talked about this. Can you tell us what sieve size you're using? I don't know the sieve size. It's just a regular kitchen sieve for tea and so on. Okay, finger or rubber gloves. Yes, I read already that. It's the same. I've been seeing. I quickly skim through the whole thing. Okay, underneath, turn the valve. Could you use a kitchen or filter paper? Again, filter paper probably not, because maybe for bacteria and so on and other smaller microorganisms, nematodes maybe, but maybe not the larger ones. I want to buy an automatic pipette. What volume range should I use? A drop of water of about 50 microliters. So I'm wondering if 10 to 100 micro. Okay, so this question here is what you want to buy a microliter pipette, and the question is which one should you buy? Honestly, it's a question of personal preference. I first bought the 1 milliliter pipette, and I found it too big. Okay, yeah. So I thought it was kind of big, right? Of course, I can also make it smaller, the volume, but I then decided I'm just gonna use the 5 to 50 microliters because and I can adjust it here anyway. So I'm perfectly happy with this one, yeah, and also the smaller tip sizes fit. Yeah, so this is these were actually quite cheap ones. Maybe not the best quality, but good enough. Yeah, good enough. And by the way, by pushing this one here, you can actually throw off the tip. Yeah, so I personally can say I'm happy with the 5 to 50 microliters. Yeah, I was actually originally thinking about getting smaller ones with yet smaller tips. But because then I figured out that you can actually make the tips smaller by pulling them out over a flame, it's not necessary to go even smaller. Yeah, and I'm not using those to actually measure out specified volumes. I'm just using those simply to move specimens around on a microscope slide. So I'm not measuring anything here. Yeah, as I understand. A Baerman funnel is used for soil. Yes, I talked about this. Yeah, the difference between 10 to 20 microliters is so little, but 20 to 200 milliliters is a huge difference. That's correct, right? Because of course you have could you spin down the sample, or would that damage the organisms? Oh, honestly, it's no problem if you spin down the sample at low speed. I've always done that. It's not a problem. Yeah, using a centrifuge. Yeah, but because many of the organisms are fairly large anyway and denser than water anyway, they sink down anyway. So if you're just patient, then it also works. Yeah. Okay. The light. I wonder why some organisms in the soil were light phobic. Oh, I already talked about that. They want to move away from light because usually there where there's light, there's also it can dry up quickly. Okay, there's some literature here. Okay, I'm just reading here. What does a water bear eat? Bacteria. Who eats water bears? I don't know. Would be interesting. I don't know. Obviously, organisms that are bigger, but I wonder what that could be. Okay. Yeah. So we talked about the size, approximately 120 microns or larger. And what time is it? Yeah, because I still want to try this other water sample here. Depends on the species. Most water bears eat the insects. Okay, eat the insides of plant cells. They drink them like juice packs. But plenty eat bacteria too, and then some will eat other microanimals. You changed the magnification? Yes, I did, but I changed it back. I hope I did. According to some images, 120 to 600 micrometers. Okay. But what do bacteria eat? They must have some sort of cellular respiration. Of course they have. Or fermentation, both, yes. So very quickly, bacteria are so-called saprotrophs. So what they have is they have extracellular digestion. They release enzymes and then they absorb the nutrients. Yeah, so obviously they do not have a digestive system, bacteria. And they absorb the nutrients and then they use cellular respiration to break down those nutrients to release energy. Yeah, so they digest the material outside of the cell and then absorb the nutrients, just like fungi, by the way. Yeah, I talked about the arrows. I designed them, and these are just pictures. Aquarium shops for valves. Okay, maybe the separating funnels that chemists use. They're made of glass, though. I'm a little bit reluctant to use glass. Yeah. About the finger gloves, yes, I already read that. And why can't we just use a funnel with a filter instead of the entire Baerman funnel? I guess you meant that you kind of dip it in a petri dish. That is indeed also possible, but I think the Baerman funnel is simply more practical if you want to go on larger scale. Yeah. So I'm quickly going to read some of your comments again here in case I skipped something over. I mentioned that I use concentrated alcohol here. Yeah, so nothing for me. And now I want to show you what I found. You won't believe it. I didn't believe it. It's I found some when I collected the moss today in the morning. Between the trees, I found the following. Let me make this higher here. I found the following. I found some plastic sticking out from beneath the leaves, and then I pulled out the plastic because I wanted to trash it. And I found this here. Look at this. There was some rainwater collected in here, and it's green. So I think this might be interesting. I don't know. Honestly, I did not look at it yet. Okay. So what I have to do is see, it's even open here. This is how the rainwater came in. Yeah. I'm just going to put it in here. The water wouldn't even flow out properly because it's all... So this is better. Still some stuff. Yeah, the biofilm would also be interesting to observe, but it's, I guess. So honestly, I have no idea what awaits us here. Okay, maybe interesting, maybe not, maybe a huge disappointment. In any case, one thing I can assure you, whatever we look at, it will probably be green. Oh, great. So, and let's give it a try. Okay, and then if this is actually something of interest, then I'll make, of course, believe it or not, a separate video of this. I don't know. Maybe some stuff is sinking to the bottom here and top. Okay. This goes into the trash. Usually standing water is never a good idea because this is a breeding ground for mosquitoes and so on. You don't want that, right? So let's have a look. And if this is something nice, Okay, then I'm happy. If not, then we've also learned something. So cover glass goes on top. Yeah, it's kind of very, very sick. Okay. So let's remove the other slide here. And where is the scope view again? Here we go. I don't like the arrow. No, no. How do you? I've got too many arrows now. Let's move it away. Here we go. And let's give it a try. No movement. Yeah, but it's green. But let's go up a little bit of the magnification here. I was actually hoping to find some I don't know, some ciliates or whatever. I don't know. Yeah, but in any case, whole bunch of algae. Green in any case, as expected. Okay. Let's go up yet further. Yeah, a bit disappointing maybe. I don't know. But you see, in any case, yeah. I don't know. Maybe if you just wait a little bit, you might actually see some oxygen bubbles forming. Yeah, because of photosynthesis. That's usually something that I would do next. I could try the following. I don't know. Usually, let's try phase contrast. Maybe we're able to see some bacteria as well. So that's now phase contrast. Yeah, usually bacteria are seen as dark structures. There are not even that many bacteria here as well. Okay. Yeah. I was actually hoping for a little bit more. But you know, that's the whole idea of microscopy. We can always, yeah, you don't always know what to expect. And maybe it's just a question of being patient, yeah? It looks like a single species of algae made its way into the bottle. That's quite well possible. Generally, it's rarely just one type of species that you find, but usually there is always some kind of a dominant one, right? So I just want to show. Let's go up. No, I cannot go higher. I can't go higher because I'm bumping. Yeah, but if you actually look here, you might actually see that some of the cells that are here, there's something moving up. At least there is in the center something moving. Yeah, not a lot. Yeah, usually there is indeed one kind of a dominant species. What surprised me a little bit is because most of the bottle was actually covered by leaves and the water was not visible, right? So only very little light seemed to have been able to enter the bottle. But at this magnification already, we can see that there are indeed a couple of different cells that are around here. Some of them are now moving. Yeah. So, yeah. So what I could do now is I'm just going to leave the jar standing, and usually you see some kind of a progression because the light is different, the temperature is different, a lot of and usually then you might see that another other organisms start to become dominant over time. Yeah. Okay, so ladies and gentlemen, I'm going to slowly think about stopping again. For those of you who joined late, a very short summary of what I've been doing. What is this? Is this some kind of a huge long fiber here? Okay, yeah. So a very short summary of what I've been demonstrating to you today. It was actually mainly two things. Over here, I showed you this so-called the Baerman funnel, right? For a semi-improvised system here that I have to collect, organisms which basically are trapped, by the water, and then they fall down and to be collected down here. And the second thing that I showed to you, which was also is work in progress, is basically the a little kind of a improvised micro manipulator, which actually basically just stays fixed over the slide, and then by moving the slide you can actually position the tip, and then you can try to suck up individual organisms this way and transfer them to different places, primarily in order to remove all of the debris and all of the unnecessary things that you do not want to have in the picture, right? In what I have done in the past very often is for individual water bears, I simply picked it up with a pipette and then transferred it. But this was also kind of a little bit, it takes a very calm hand. Yeah. So. What I'm going to do now is I'm going to call it a day again. I'm going to call it quits. All of these things are, of course, work in progress, so I'll report back to you on more or less how successful I was. As always, I'm working on again on some YouTube videos for this channel here. If you like these type of things, please, I would like to invite you to subscribe. That's always important, not only to my channel, but generally to all channels that are microscopy related. Let's support the community a little bit here. And if you have any suggestions or wishes, then please do comment on them or post them, and I'll try to follow up on those. Yeah. Quick question: any microscopy ideas with bananas? Yes, of course. What do you do? Banana. I can quickly tell you. You take a very soft banana, a small piece, and you add a little bit of iodine to the banana. Also works without iodine, right? And you squeeze the banana between the cover glass and the microscope slide. You put some tissue paper on, you squeeze it, and you're going to see the banana starch grains. Okay? The banana starch grains are quite nice to look at. And you know what? I actually will show you something. Just a second. Just a second. Nah, I don't have it here. Where is it? Okay, no, unfortunately, I don't have it here right now. But essentially, there is I don't have the picture. I actually made a picture of these banana starch grains once, and they actually are quite nicely visible. Okay, works with and without iodine. Okay, But you have no iodine, then do it without iodine. You are still able to see those oval starch grains inside the bananas. Yeah, so that's quite easily possible. Yeah. Time to get some iodine, or maybe you go simply to the drugstore. Sometimes they also sell iodine for disinfecting. Just make sure that the iodine does not contact any microscope parts. Iodine is very aggressive. You might want to dilute it a little bit. Yeah, but honestly, I will take this as an idea, and then I will show you some starch grains next week. How is that? Okay, we can look at some starch grains from different sources with or without iodine. Okay, I think I'm going to call it quits for the day. Hope you liked the stream. Thank you that you joined again. Happy microbe hunting as always, and see you around. Bye bye.